Well, hello, and welcome to 7.2 Cell Structure. Second lesson in Chapter 7, obviously. On the far side's a little funny. Singled cell bars. Ha, ha, ha. Get it? So our goal is to compare and contrast the general structures of plant and animal cells, and also to be able to compare and contrast the general structures of prokaryotic and eukaryotic cells. So let's think about it. At first glance, if you happen to go into a factory can be a very puzzling and confusing place. And the sheer diversity of the activity can be awfully confusing. However, if you take time and you watch carefully, what might at first seem like chaos begins to make sense. And guess what? The same is true for the living cell. It is like a factory, and you're going to need to take your time and make careful observations with this lesson. So we have several objectives. The first one is describe the structure and function of the cell nucleus, describe the role of vacuoles, lysosomes, and cytoskeleton, identify the role of ribosomes, endoplasmic reticulum, and Golgi Apparatus making proteins, describe the function of the chloroplasts and mitochondria in the cell, and also describe the function of the cell membrane. All right, objective 1, structure and function of the cell nucleus. In the same way that the main office controls a large factory, the nucleus is the control center of the cell. The nucleus contains nearly all the cell's DNA, and with it, the coded instructions for making proteins and other important molecules that we need. The nucleus is surrounded by a nuclear envelope, call this the nuclear envelope, all right, which is composed of two membranes, the inner and the outer membranes. The nuclear envelope is dotted with thousands of these little nuclear pores, which allow material to move into and out of the nucleus. Like messages, instructions, and blueprints moving in and out of a main office, and the steady stream of proteins, RNA, and other molecules move through the nuclear pores to and from the rest of the cell. Chromosomes contain the genetic information that is passed from one generation of cells to the next. Most of the time, the thread-like chromosomes are spread throughout the nucleus like spaghetti, and we call them in the form of chromatin, all right, which is a complex of DNA bound to proteins. So when a cell divides, its chromosomes condense and then can be seen under a microscope. That's when you see it looking like this with the famous X figure. Most nuclei also contain a small, dense region known as the nucleolus. The nucleolus is where the assembly of ribosomes begin. So objective 1 is to describe the structure and function of the cell nucleus. The nucleus contains nearly all of the cell's DNA, and with it the coded instructions for making proteins and other important molecules. All right, and moving on. Objective 2 is to describe the role of vacuoles, lysosomes, and the cytoskeleton. So in a eukaryotic cell, can be divided into two major parts. First, you have the nucleus, and then the rest of it, which is called the cytoplasm. And within the cytoplasm, there's numerous organelles, which we're going to learn about. But let me tell you a little bit about the cytoplasm first. And the cytoplasm is the fluid portion of the cell outside the nucleus, and the cytoplasm of the cell actually can move itself. And it's called actually cytoplasm streaming. It's the back and forth motion of the cytoplasm. And there are two different types or consistencies of cytoplasm within the cell. The first is the gel state, which is on the outer side. It's firmer and found around the outer edge of the cell. The other type of cytoplasm is the sol state, which is more fluid and found towards the interior of the cell, more toward the nucleus. Prokaryotic cells have cytoplasm as well, even though they do not have a bound DNA in a nucleus. So many cellular structures act as if they are specialized organs, and these organs are known as organelles, and that word literally means little organs. So understand what each organelle does helps us understand the cell as a whole. So let's move on. You have the vacuoles and you have the vesicles within the cytoplasm. Many cells contain these large sac-like membrane structures called vacuoles, as you can see here. And these things are there to store materials such as water, salts, proteins, and carbohydrates. In many plant cells, there is a single large vacuole filled with liquid. The pressure of the central vacuole in these cells increases the rigidity, making it possible for plants to be able to support heavy structures such as leaves and flowers. So when the water goes into the vacuole, it expands, makes push pressure against the cell membrane and then the cell wall, and it makes the plant stand up nice, straight, and tall. Cool, huh? Vacuoles are also found in some unicellular organisms and in some animal cells, but they're generally much smaller. For example, the paramecium contains an organelle called a contractile vacuole, and by contracting rhythmically, this specialized vacuole pumps excess water out of the cell. All right, nearly all eukaryotic cells contain smaller membrane-enclosed structures called vesicles. So now we're talking about all plants and animal cells. Vesicles are used to store and move materials between cell organelles as well as to and from the cell surface. So you think about that word vesicles, it sounds like vessels, doesn't it? And yes, that's what they do. They move things around. Temporarily storage them and then moving them around. There's some little pictures of some little vesicles. So lysosomes are next. Lysosomes are kind of cool. They are small organelles filled with enzymes that function as the cell's cleanup crew. The lysosomes perform the vital function of actually removing junk that might otherwise accumulate and clutter up the cell. One function of the lysosomes is the breakdown of lipids, carbohydrates, and proteins into small molecules that can be used by the rest of the cell. So basically, you might say they digest those macromolecules. Lysosomes are also involved in breaking down other organelles that have outlived their usefulness. So maybe they're just not functioning properly, and the lysosomes can go along and eat them up. Biologists once thought the lysosomes were only found in animal cells, but now it's clear that lysosomes are also found in a few specialized types of plant cells also. So, moving on to the cytoskeleton, and yes, it's pretty much what the word sounds like. All right, eukaryotic cells are given their shape and internal organization by a network of protein filaments known as the cytoskeleton. Yeah, it works as a scaffolding to support the cell and organelles, and also acts like a highway to bring information within the cell. I'll tell you what I mean by that in a second. Certain parts of the cytoskeleton also help to transport materials between different parts of the cell. So you can see this picture right here. These are microtubules, and on them they have these little proteins, and they're being transferred along to other parts of the cell. That's what it means. It's like a conveyor belt that carries materials from one part of the factory to another. Microfilaments, all right, and microtubules are the two of the principal protein filaments that make up the cytoskeleton. So what I'm trying to say there is within the cytoskeleton there are two types of cytoskeleton, microfilaments and microtubules. Let's look at the microfilaments first. Microfilaments are thread-like structures made up of protein called actin. They form an extensive network in some cells and produce a tough, flexible framework that supports the cell. They also help the cell to move in some cases, that they can contract like this. So microfilament assembly and disassembly is responsible for the cytoplasmic movements that allow cells like amoebas to crawl along the surfaces. Moving on to the microtubule part of the cytoskeleton then. Microtubules are hollow structures, they're like these things right here, all right, made up of proteins known as tubulins. So tubules are made of tubulins, and they play a critical role in maintaining the cell shape. Microtubules are also important in cell division, where they form a structure known as the mitotic spindle, which you'll learn about when we get to mitosis, which helps us to separate the chromosomes. On page 355 and 356 in your Middle of Bean book, there's a little more information too. Microtubules help to build projections from the cell surface too. These little projections are known as like cilia, all right? And if it's a long tail-like thing, it's la flagella. And so microtubules enable the cells to swim rapidly through liquids too. They are arranged inside the microtubule. There's a 9x2 pattern inside there. And small cross bridges between the microtubules in these organelles use chemical energy to pull on or slide along the microtubules, allowing the cells to produce controlled movements too. So they're both about support and helping the cell to move, and the microtubules, the difference there is that they make up the little cilia and the flagella. All right. Microtubules also in animal cells, make up the structures known as centrioles. All right. Centrioles are located near the nucleus for a reason, because they help to organize cell division. And we'll learn more about those too when we go to mitosis also. Centrioles are not found in plant cells though, only in the animals. So let's just recap here. All right. So microtubules form the cilia, flagella, and the centrioles, and they help maintain the cell shape. Microfilaments also help maintain the cell shape too. All right. So objective 2 is to describe the role of vacuoles, lysosomes, and cytoskeletons. Vacuoles store materials like water, salts, proteins, and carbohydrates. Lysosomes are there to break down lipids, carbohydrates, and proteins into small molecules that can be used by the rest of the cell, and they're also useful in breaking down the other organelles that are not useful anymore. And the cytoskeleton helps the cell maintain its shape and is also involved in the movement, and the cytoskeleton is composed of microtubules and microfilaments. Objective 3, identify the role of ribosomes, endoplasmic reticulum, and the Golgi apparatus in making proteins. So let's move in. Cells need to build new molecules all the time, especially those wonderful proteins. And as you know, enzymes are made up of proteins, so proteins catalyze chemical reactions and make up important structures in the cell. And because proteins carry out so many of these essential functions of living things, a big part of the cell is devoted to their production and distribution. So proteins are synthesized, basically made, on ribosomes, sometimes in association with the rough endoplasmic reticulum in eukaryotes. I know I just said some words there that you don't know what they are, but we'll get to them. Ribosomes are small particles of RNA and protein found throughout the cytoplasm in all cells. Ribosomes produce proteins by following coded instructions that come from the DNA. Each ribosome is like a small machine in a factory, turning out proteins on orders that come from its DNA boss. In eukaryotic cells, contain an internal membrane system known as the Endoplasmic Reticulum, and we just like to call it the ER. The endoplasmic reticulum is where lipid components of the cell membrane are assembled, along with proteins and other materials that are exported from the cell. The portion of the ER, involved in the synthesis, making of proteins, is called the rough endoplasmic reticulum. It is given its name because of the ribosomes found on the surface. Little ribosomes are bumped all over the surface of the ER there. So it's rough. That's why it's called rough. The newly made proteins leave these ribosomes and are inserted into the rough ER, where they may be chemically modified in there. So you can look at this process of what happens also on page 358 in your little baby book too. A little video there for you guys. So moving on, the other portion of the ER is known as the smooth endoplasmic reticulum because ribosomes are not found on its surface. In many cells, the smooth ER contains collections of enzymes that perform specialized tasks, including the synthesis of membrane lipids. We need lipids for our cell membrane, which we'll get to that later also, and the detoxification of drugs. All right. Proteins produced in the rough ER move next to what's called the Golgi Apparatus. The Golgi Apparatus, which appears as a stack of like flattened pancakes like that, these proteins are bundled in these tiny vesicles from the rough ER, and they go out and they go to the Golgi, and the Golgi brings them in. And they carry them to the Golgi Apparatus. The Golgi Apparatus will then modify, sort, and package the proteins and other materials from the ER for storage in the cell or released outside the cell. It's something like a customization shop where the finishing touches are put down on proteins before they're ready to leave the factory. So again, the Golgi Apparatus, then proteins are shipped to their final destination inside or outside the cell. It can be pushed out or used inside the cell. So objective 3 was to identify the role of ribosomes, endoplasmic reticulum, and the Golgi apparatus in making proteins. So the proteins are assembled on the ribosomes. Proteins made on the rough endoplasmic reticulum, or ER, include those that will be released or secreted from the cell, as well as many membrane proteins and proteins destined for lysosomes and other specialized locations within the cell. The Golgi apparatus modifies, sorts, and packages proteins and other materials from the ER for storage in the cell or release outside the cell. Objective 4. Describe the function of chloroplasts and mitochondria in the cell. Okay, this is my favorite part actually. Organelles that capture and release energy. These are what they do, chloroplasts and mitochondria. So you all know that all living things require a source of energy. Most cells are powered by food molecules that are built using energy from the sun because we eat it from the plants, right? Chloroplasts and mitochondria are both involved in energy conversion processes within the cell. So mitochondria and chloroplasts. And I want you to note that plant cells also have mitochondria. Plants and some other organisms contain chloroplast. Chloroplasts are the biological equivalent of like little solar power plants. They capture the energy from sunlight and convert it into food that contains chemical energy in a process called photosynthesis. Inside each chloroplast, the chloroplast is constructed of two membranes, outside membrane and inside membrane. And inside these organelles of these chloroplasts are large stacks of other membranes called stromas, but we'll learn about those when we get to photosynthesis, which contain the green pigment chlorophyll. So, nearly all eukaryotic cells, including plants, contain mitochondria. Mitochondria are the power plants of the cell. They convert the chemical energy stored in food into compounds that are more convenient for the cell to use, and that's called cellular respiration. The mitochondria is composed of two membranes. It has an outer membrane, and then this inner membrane, which is all folded up inside the organelle. One of the most interesting aspects of mitochondria is the way in which they are inherited. In humans, all or nearly all of our mitochondria come from the cytoplasm of the ovum or egg cell. So basically, you get your mitochondria from your mother. So go thank your mother right now. So, chloroplasts and mitochondria contain their own genetic information in the form of small DNA molecules, which is really quite fascinating. They have their own DNA, chloroplasts and mitochondria. Why would that be when you have a nucleus full of DNA? Well, there's a theory about that, and it's called the endosymbiotic theory. And it suggests that the chloroplasts and mitochondria are descended from independent microorganisms. Way millions of years ago, these independent microorganisms were engulfed by the cell that we know, and that's how the mitochondria and the chloroplast became part of the eukaryotic cells that we know today. They were kind of eaten. I guess they needed it. Okay, so objective 4, describe the function of the cell membrane. Actually, describe the function of chloroplasts and mitochondria in the cell. Let's recap here. Chloroplasts capture the energy from sunlight and convert it into food that contains chemical energy in a process called photosynthesis. Mitochondria convert the chemical energy stored in food into compounds that are more convenient for the cells to use in a process called cellular respiration. Yes. All right. Now, Objective 5. Describe the function of the cell membrane. All right, cellular boundaries. Like a working factory has walls and a roof to protect it from the environment outside, and also to serve as barrier that keeps its products safe and secure until they're ready to be shipped out. Similarly, cells are surrounded by a barrier known as the cell membrane. Many cells, including most prokaryotes, also produce a strong supporting layer around the membrane known as a cell wall. The main function of the cell wall is to provide support and protection for the cell. Makes sense. That's what walls usually do. provide support and protection. Prokaryotes, plants, algae, fungi have cell walls. Many prokaryotes, I should just say all. Animal cells do not have cell walls. Cell walls lie outside the membrane, and most are porous enough to allow water, oxygen, carbon dioxide, and certain other substances to pass through them very easily. So, all cells contain a cell membrane that regulates what enters and leaves the cell and also protects and supports the cell. The cell membrane helps the cell keep out the bad stuff because it has a cell recognition structure. It's like a self, not self thing going on. It says, okay, you're not one of us, so you're not allowed in. All right? So the cell membrane is not arranged in any beautifully symmetrical order or anything, no orderly manner. The atoms are just added and removed as needed. It's like a living entity. Things come and go. Proteins and carbohydrate chains and all kinds of stuff moves in and out. And they're just all over the place. The composition of nearly all cell membranes is a double-layered sheet called a lipid bilayer, which gives the cell membranes a flexible structure and forms a strong barrier between the cell and its surroundings, and is made of what's called phospholipids. And this right now I'm telling you is incorrect. It's a hydrophilic head, not a hydrophilic head. What the heck is that? Hydrophobic and hydrophilic is what it should write right there. So the composition, I'll just that. So as we learned in chapter 2, many lipids have oily, fatty chains attached to them, to chemical groups that interact strongly with water. The fatty acids portions of such a lipid are hydrophobic down here, water-hating, while the opposite end of the molecule is hydrophilic, water-loving. That is wrong, hydrophilic. When such lipids are mixed with water, their hydrophobic fatty acid tails cluster together, because they don't want to be around the water, while their whole hydrophilic heads are attracted to the water. As a result, you can see there's a lipid bilayer. You have some on this end, heads on the outside of the water with the inside lipids, and some on this end. So you have them inside and outside of the cell. A lipid bilayer is the result. Okay. Because most cell membranes contain protein molecules that are embedded in the lipid bilayer, protein channels here, here, and here. Carbohydrate molecules are attached to many of these proteins. Because the proteins embedded in the lipid bilayer can move around and float around among the lipids, and because so many different kinds of molecules make up the cell membrane, scientists describe the cell membrane as a fluid mosaic. So some of the proteins form channels and pumps that help move the materials across the cell. Here's an example of a protein pump. Many of our cell membrane, I should say, many of the carbohydrate molecules act like chemical identification cards. They have a carbohydrate chain on them, allowing individual cells to identify one another. Although many substances can cross biological membranes, these membranes, some are too large or too strongly charged to cross the lipid bilayer. So if a substance, that's not right. If a substance is able to cross a membrane, the membrane is said to be permeable to it. A membrane is impermeable to a substance that cannot pass across it. Most biological membranes are selectively permeable, and we're going to learn about osmosis and diffusion in a couple of chapters. Meaning that, I think it's next chapter, meaning that some substances can pass across them and others cannot. Selectively permeable membranes are also called semi-permeable membranes. And there's a function, maybe you go to my content area where the PowerPoints are at, and you can click on this and watch the cell function video too. Here's a recap of all, this is a great resource for you to use, all the different types of organelles within each plant and animal cell. And I just want to talk to you one more thing. Why are cells so small? Okay, the reason is because of the cell membranes. The more membrane surface area gives better control rather than one large cell. So if you have one large cell, you have less control over all the organelles and everything. That's why cells divide, because they get too large and they need to divide. We'll learn about that more too. So as the cell size increases, the membrane gets smaller compared to the cell volume. So they need to keep themselves small enough in order to control everything that goes in and out of other thing. Otherwise, things get out of control. So objective 5 is describe the function of the cell membrane. The cell membrane regulates what enters and leaves the cell and also protects and supports the cell. Objective review. Let's ask yourself, can you describe the structure and function of the cell nucleus? Can you describe the role of vacuoles, lysosomes, and cytoskeleton? Can you identify the role of ribosomes, endoplasmic reticulum, and the Golgi apparatus in making proteins? Can you describe the function of chloroplast and mitochondria in the cell? And finally, can you describe the function of the cell membrane? I hope so. And there's a funky picture for you to look at this time. Makes it moving. All right. Bye.